Okay, so this presentation is about nervous system cells and organization, just of the nervous system in general. Looking at the nervous system and its general organization and the cells that are involved, we can see that we have several functions of the nervous system that we want to look at. First off, we have sensory input. And sensory input, we gather information, we monitor changes that occur both inside and outside the body, and the changes that we're looking at are referred to as stimuli. We take that information and we integrate it. Basically, we've got a whole different set of nerves called interneurons that just process this information and decide whether it is important enough for us to act on. Now, beyond that, what we look at is, we have motor output. A response, basically, to the integration aspect is the motor output. And that can go to muscles or glands, just determines on what kind of activity we've got going on. Now, when we look at this, and this chart actually should be fairly familiar to you. I think in chapter 1 we talked about feedback loops, and so sensory input goes in, we have the processing area, we have motor output that goes out. Now, once we've done that, what we can look at is just general structural classification of the nervous system. We have the central nervous system, which consists of the brain and the spinal cord, and we have the peripheral nervous system, which are nerves outside of the brain and spinal cord. These are spinal nerves and cranial nerves. Simple way to look at this, I think, is that things that are surrounded by bone, like the skull or the spinal column, what we're looking at there, that's the central nervous system. Anything that goes outside of those, that is our peripheral nervous system. Again, what we're looking at is sensory, which is also known as the afferent. And I'm not likely to ask you about afferent and efferent, but afferent is coming in, and we have nerve fibers that carry this information to the central nervous system, and then motor fibers, or efferent, which carries information away from the central nervous system. Now, basically what this looks like graphically is we have the central nervous system here, which is the brain and spinal cord, and from sense organs going through sensory nerves, which are in the peripheral nervous system, we get that information to go to the central nervous system. Central nervous system processes that information, sends a signal out to the peripheral nervous system. Well, it does so as motor, which can be somatic, that is skeletal muscles. Somatic system is voluntary. It is what we can control. And we also have the autonomic. Now, when I think autonomic, I really think automatic, because this is stuff that we really don't think about very much at all. And we've got the parasympathetic and the sympathetic that we can look at there. Parasympathetic is sort of what we refer to as the rest and digest, and the sympathetic speeds us up. When we're looking at the sympathetic, that energizes us, activates us. The parasympathetic slows us down. So through the course of the day, basically we have either a foot on the gas or a foot on the brake, and it is sort of a balance between those two that keep us going. When we look at peripheral classification, or functional classification in the peripheral nervous system, again we have these two subdivisions: somatic, which is voluntary, and autonomic, which is involuntary. We also have a whole lot of cells that support our nervous system. Our nervous system, very important. That's where our consciousness resides. It's where all our control resides. And so we have to take very good care of it. And we end up with these support cells that are referred to as glia. Now, The first computers, they were massive things. They were like a city block and 4 stories tall, and they had hundreds of little technicians running around making sure everything worked properly. You can kind of think of the Glia as being like that. I also think of them in terms of NASCAR. My wife used to love NASCAR racing, and I still don't understand that, because basically it's just a bunch of guys turning left all day. But the thing is that it's more than just the guy in the car. You have a whole pit crew that takes care of the racer. You have someone who puts fuel in. We have people who repair the car. We have people who get fluids to the driver and so forth. And that's what a lot of the Glia does for us. Now, the most abundant cells that we deal with are the astrocytes. And they're called astrocytes because they are star-shaped, like astral. And they help brace neurons. Beyond that, they also form a barrier between capillaries and neurons. They control what goes into the neurons. It's, again, these are very important cells, and we don't want toxins to get to them. We don't want other substances that could cause harm. So we have these astrocytes that control it. I think sort of the astrocytes as being like waiters. You don't go into a restaurant, go back to the kitchen, prepare your own food, and bring it out. You have someone who goes and gets it for you, selects what goes in there, and then gets it to you. So they also control the chemical environment of the brain. As we look at these nervous tissue support cells, this is what an astrocyte pretty much looks like. It's got processes that go off and surround capillary beds, which select what nutrients come in, what substances pass through, and feed that to the neuron. Now, these are not completely selective, but they make up a very important part of the blood-brain barrier. Now, they can block some substances from coming through, others they can't. Oxygen, carbon dioxide pass through regardless of whether it's astrocytes there or not. Some toxins will pass through, like alcohol, but other substances won't. Antibiotics actually don't get past the blood-brain barrier, which does mean that if you've got an infection in the brain, it can be particularly difficult to treat. Another supporting cell that we have is the microglia, and these are kind of spider-like in shape. They're phagocytic, that is, they go through, they clean up debris, they just generally are the maintenance crew of the brain and the central nervous system. And they look kind of spider-like. In fact, they are mobile, so these are crawling around inside your brain, constantly cleaning stuff up. So yes, you have a whole bunch of spiders in your brain crawling around. Yay. We have ependymal cells, and these line cavities in the brain, they create cerebral spinal fluid and circulate that. And these cells to me look a whole lot like pseudostratified ciliated columnar epithelium, in that they generate this fluid and the cilia move the fluid around. Okay. Next cell we're looking at, these are oligodendrocytes, and oligodendrocytes make myelin. Now, myelin's very important in terms of increasing nerve signal speed. So, oligodendrocytes allow nerves to send signals fairly rapidly. These are located in the central nervous system. We have a similar cell in the peripheral nervous system called a Schwann cell that also makes myelin, but they make them really, really differently. Now, looking here, this is an oligodendrocyte. It has processes that go off and wrap around different nerve cells. These do not wrap those nerve cells very tightly, and as a result, damage in the central nervous system doesn't heal very well. If you were to cut an axon in the central nervous system, that portion of that axon would die off, and this would collapse, which would prevent the axon from being able to grow back through. It's a little bit different in the peripheral nervous system. Now, the cells that we've looked at thus far, these are central nervous system cells. Now we're looking at support cells that are in the peripheral nervous system. We have satellite cells, which protect neuron cell bodies. They kind of act like the microglia, oligodendrocytes, all of that together. They keep everything safe. And then we have Schwann cells, which make the myelin sheath in the peripheral system. Now, Schwann cells, they actually cover about 1 millimeter of an axon in the central nervous system. And we have axons that go all the way from our brain down to your big toe. So we have some axons that are, 2 meters long. That means that for a cell like that, we would have, 100, 200 separate Schwann cells, which does come at a big expense to the body. There's a lot of energy in maintaining this. Conditions that can occur when this breaks down, you can have breakdown in the central nervous system. Basically, you get multiple sclerosis. If we're looking at that in the central or peripheral nervous system, what we're looking at is Lou Gehrig's disease, or ASL. So these are very important to the functioning of nerve cells. But again, not every nerve is covered with Schwann cells. There is a big expense associated with maintaining these. So if we don't need to be able to send a signal really fast, we're not going to put myelin on it. So we want this on certain pain receptors and on things that control muscles, so we can react quickly, and so that if we get something that's doing damage to us, we can stop before it takes very long. This allows nerve signals to travel at about 200 meters per second. On the other hand, if you're looking at cells that don't have myelin, they send signals at about 2 meters per second. So you can see there's about 100-fold difference in the rate that signals are sent. Cells that don't have myelin are going to be in tissues that really don't have to send signals quickly. So if I'm eating something, I don't have to be digesting my food the second that it hits my stomach. So we're going to send a signal on the slow cells, which will result in, a second or two delay in response. So